Good morning, good afternoon, good evening, everyone. Depends on where you are at. Welcome to today's webinar. I'm Chiming Wang. I'm the marketing director at Cyagen. Joining me today is our senior scientific BD manager, Dr. Joseph Wekselblatt. Joe is an experienced scientist who has studied visual capabilities in different species for over a decade. He received his Ph.D. in neurobiology from the University of Oregon. After completing his Ph.D., he went on working as a postdoc researcher at HHMI investigator Dr. Shapiro's lab at Caltech until 2023, when he joined Cyagen as a senior scientific BD manager. The title of Dr. Wekselblatt's talk is Accelerating Glaucoma Drug Discovery from Preclinical Models to Efficacy Validation. During his presentation, please feel free to type in your questions in the chat box. We will have Dr. Wekselblatt answer as many questions as possible. Joe, I'd like to hand it over to you.
Thank you so much, Ximing. It's very nice to be here and to share with you Cyagen's capabilities. So I'll just take a moment to get the slideshow up. Perfect. Well, yeah, thank you so much, Ximing. I'm Joseph Wekselblatt. I've been working for Cyagen for a little over 2 years now, and I have a background in both neuroscience and ophthalmology. So it's my pleasure to be at this webinar and share with you the latest information on translating glaucoma models to accelerate ophthalmic therapeutic discovery. So let's start with why glaucoma remains such a major unmet need. I've divided the talk into 4 sections. First is just a background on glaucoma. Second, I'll introduce you to some of the current animal models that we have for glaucoma. And then third, I'll talk a little bit about the application of these animal models in primary research. And then at the end, I'll introduce Cyagen services for our CRO platform and show some ways that we can help accelerate your research. Okay, so glaucoma is one of the biggest causes of blindness, both in the United States and globally. And as a homage to the Halloween season, we've called this the section on the vision killer. It's a chronic progressive disease. It's thought to affect up to 350 million people globally by 2050. So it's really an unmet need and one we think we're poised to start to solve. So glaucoma is basically characterized by a pathological elevation in intraocular pressure, which causes stress on the retinal ganglion cells. And then after they die, the optic nerve atrophies and you start to have effects in the visual field. And the exact pathogenesis remains unclear, but the only current treatment is to lower the intraocular pressure. And many research directions are starting to target both the production of aqueous humor and the drainage of it as alternate methods. The aqueous humor is produced, it's like the fluid that's inside the eye. And so understanding the fluid dynamics of the eye is essential to understand how we should use this model to inform our drug discovery. So when the aqueous outflow, let me see if I can get my laser. It's produced here by the ciliary bodies, and it kind of flows into the anterior chamber and then flows out through the tubicular meshwork and what's called the canal of Schlemm. That's the main pathway. And so when this is obstructed, either one, the main or the secondary pathway, you can get a buildup of aqueous humor in the anterior chamber, which causes increase in your ocular pressure and thereby puts stress on the retina. So let's see. The main kind of glaucoma is called primary glaucoma. There's also secondary and congenital, but I'll focus on primary, one, because it's the most common, and two, because we have the best understanding of the dynamics, the fluid dynamics that cause it. So in open angle glaucoma, the increased interocular pressure is due to the trabecular meshwork and the island of Schlemm not working right. And the angle closure glaucoma here is caused by a pupillary block. And so the interocular pressure stems from decrease in outflow in the interior chamber. The location of the outflow determines the kind, open or closed angle. And here I just want to review some of the risk factors, mainly age, family history, and then measured increase in interocular pressure. There's also some conditions that are comorbid and infectious diseases that predict onsets of glaucoma, and some medications to avoid if you do have glaucoma. And then the symptoms and severity are related to the type of glaucoma. So the open angle primary glaucoma tends to have symptoms like headaches at the start, and then start to have bilateral field narrowing and night blindness, where the angle closure one, patients often experience really bad headaches, eye swelling, and then sudden vision loss. Um, so yeah, finally here I want to review the common drugs that are currently approved and used for glaucoma treatment. I'll go through these briefly, but most of them are aimed at either promoting the outflow of the aqueous by reducing swelling, or to reduce the production of the aqueous humor. And so, yeah, the current glaucoma treatments tend to involve drugs or surgery to regulate either production or outflow, thereby controlling interocular pressure. And I think a little later I'll show some new small molecule drugs and AAV gene therapies that are kind of part of the next generation of treatments for glaucoma. Okay, so now I think I'd like to take a look at how different animal models can help us replicate the disease mechanisms in humans, and inform our treatment methodology, and also the choice of treatment for individual patients. And of course, there's no perfect animal model for glaucoma, but I think among the variety of them, each has its own kind of benefits, and can inform our preclinical research. So here I'm kind of summarizing some of the animals that are used. This isn't an exhaustive list, and I think I'll focus mainly on the rodent models, since those are the ones most used in the preclinical space, and also the ones we're providing to our clients. So we have a couple of genetic models. These are spontaneous models of glaucoma. And then we have several inductive methods where we're artificially increasing the interocular pressure by means of magnetic beads, or a couple other methods I'll review on the next slide. And so Cyagen offers both mice and rat models. I think there's advantages to both. The rats have bigger eyes, and so the surgeries are a little easier, but neither have maculas. So for macular degeneration, we tend to work in collaboration with primate labs. For glaucoma specifically, we offer 4 inductive methods. Magnetic beads, which I mentioned briefly, and so that's basically an injection um, of beads that kind of form a circle around the sclera and don't let the aqueous humor drain. We have an optic nerve crush injury where we're modeling damage to the retinal ganglion cells, and we can assess the health and the regeneration of those axons after injury. We have a ischemic reperfusion model where we increase the intraocular pressure by injecting into the anterior chamber. And finally, an NMDA-induced exotoxic damage model. And so the NMDA causes hyperexcitation, which causes cell death in the retinal ganglion cell layers. So once we establish a model, we aim to quantify the efficacy using multimodal in vivo readouts. So for example, with the magnetic bead model, we can inject in one eye and assess against the control eye in the same animal. And you can see here at day 0 that the eye is healthy. With the magnetic beads, you can see that they form a ring around the sclera here. And 10 days in, you can see some of the damaging effects of the increased ocular pressure. And here we're measuring, and we can see that the intraocular pressure almost doubles, and that stays high for up to 3 weeks. Okay, this second model is the optic nerve crush, and you can see, as is schematized here, we can kind of take pincers and crush the nerve, which causes death of some of these axons that originate in the retinal ganglion cell layer and go to the brain. And so here, the flash ERG is a physiological measurement of the activity of those RGCs. And we can see with the optic nerve crush, you have quite a reduced amplitude in both the A wave and the B wave. And here in the flash VEP, it's almost completely flat. We also performed pathological tests in histology using immunofluorescence. And you can see here the TUBB3 stain, which is pretty specific for axons of the retinal ganglion cells along the optic nerve. You can see here the axons are like healthy looking, and there's a lot of them. And after optic nerve crush, you see that a lot of those have retracted, or the density's gone down. And so this is a nice model to then perform a therapeutic treatment on. Okay, so third model I had mentioned is the ischemia reperfusion model here. So you can see that in this model, we're accessing the anterior chamber and increasing the intraocular pressure by injecting fluid. And after the ischemic reperfusion, you can see blanching of the iris and the retina. And the severity of that, can be observed in real time, and is dependent on the pressure or the height of the reperfusion liquid. When you have that increase in intraocular pressure, you get a kind of decrease of blood flow into the retina, which causes that ischemic injury. So here again, I'm showing the response to the ERG, and you can see the control here in blue, and then different days after the ischemic injury. So red is 3 days after, 7 days after you see in green, and 14 days after you almost see an abolishment of the B wave there. And you see the same pattern on the right here for the A wave. So it got that chronic progressive feel that we see in glaucoma. Over here, you can see the retinal ganglion cell death by different area of the retina. So we have central on top, the kind of intermediate layer, and the peripheral layer on the bottom here. And you can see here the effect seems strongest in the peripheral retina. We can also measure retinal thickness, and we can do that layer by layer. You can see schematized here by day, you get a decrease in the thickness of the retina over time, all of which are indicators that we can use therapeutic candidates to assess the protective nature of them against this glaucoma injury. Okay, so the last model, the last inductive model, at least I want to introduce, is this NMDA-induced glaucoma. Um, so this is performed with an intravitreal injection of the amino acid glutamate or its analog NMDA, and can cause cell death without affecting the intraocular pressure. Glutamate's a major excitatory neurotransmitter in the central nervous system, and it's expressed in bipolar cells, photoreceptors, and ganglion cells. And so in glaucoma development, we see excess of glutamate leads to overstimulation of the NMDA pathways, and we think is one of the reasons that the RGCs ultimately die. So in this model, we found no real change in retinal thickness, but you do see a progressive cell death of the retinal ganglion cell layers, and that's also dose dependent, which tends to be a great feature of the model. So we can test different severities of glaucoma, for example. So you see this dose-dependent nature here in the physiological measurements, especially in the B wave, where you have the 2 nanomolar drug and the 20 nanomolar drug in green. Okay, so now I want to show how these models are applied to test different therapeutic modalities. And for that, I think I'll briefly introduce a few papers that used animal models and different therapeutic approaches to evaluate the drug efficacy. In this first example, the authors used an AAV drug to target the aqueous production. Um, so they evaluated this in 2 different animal models, both the magnetic beads as well as genetic animal model I mentioned earlier, the DBA-J2 mice that are genetically predisposed to getting glaucoma. And here efficacy was monitored using indicators like intraocular pressure measurements, ERG, and retinal thickness. And they also did some targeted protein expression. So in this case, the therapy was quite effective. You can see the reduction in intraocular pressure here. And this kind of illuminates the new approaches, using AAV to target a specific gene known to be involved in aqueous production in the ciliary body. So this was exciting for us to see. A second study, which also used AAV, this time to regulate 2 other genes, which are known to be involved in cell death. So here the authors employed the optic nerve crush model and also a second model called silicon oil-induced hypertension, which is it's very much like the magnetic beads model, but actually silicon oil has the advantage that it's reversible. You can take it out of the anterior chamber. But anyway, so they use that and the optic nerve crush models to evaluate the health of the RGCs, both in the presence of the AAV and without it. And so they targeted 2 different genes, the CHOP gene and the ATP4 gene. And in both cases, they saw a huge increase in the survival rate. And when applied together, they see that there's a synergistic enhancement of survival of the RGCs almost up to 100%. So here we see, beyond controlling the pressure, neuroprotection is a really important pathway for drug discovery kind of going forward. Um, Other than AAV, I think small molecule drugs have been really effective in the glaucoma space and in other places in ophthalmology. So this study is a small molecule drug using the NMDA-induced model. And they had two different candidates, both which showed promising results. You can see here the retinal ganglion cell layer. It seems to be much more preserved in the case where they give NMDA and the small molecule drug versus NMDA and the DMSO. So this small molecule drug targets the RIP pathway, which is one of the pathways that inhibition of it leads to neuroprotection. It's a necrotic pathway. Um, so even microRNAs and exosomes are now entering the field, and I just wanted to quickly introduce a recent study using microRNA-based therapy. And in this case, they're treating the ischemia reperfusion model. Their drug is here, the 21,5P. And you can see, oh, it's kind of small here, but it serves as a really nice anti-inflammatory factor and causes the retinal ganglion cells to look almost like the control after the reperfusion. So yeah, we're excited about these new advancements in the field, and we're very happy to offer these animal models if it serves your needs. So please feel free to inquire about any of these. Um, I want to talk a little bit about our capabilities in being able to serve the community, both as a CRO, a contract research organization, so we can help with piloting studies. We can actually do full studies end to end, and one of our main service areas is ophthalmology. So yeah, we're very happy to help support your research at any stage. Um, we tend to serve, comprehensive set of offerings in the preclinical space. I wanted to highlight two. We have our own AAV capsid production facility and R&D team who's making AI-enabled AAVs with enhanced penetrance of the retinal ganglion cell layers and the whole eye. And also our in vivo efficacy platform where, we're able to test animals for a whole slew of electrophysiological, behavioral, and other needs. You can look at the tools and technologies that make this possible here. So we have everything from the ERG machine to the slit lamp scope, which I'll talk a little bit about later, and then a whole slew of behavioral tasks. So we have optokinetic reflex, we have Morris water maze, we have light dark box. And so, yeah, we can cover detection, everywhere from fundus imaging to behavior. And here I have in pink indicating the correspondence of the types of detections for each of the machines we have here. So we're also adding to our facility, and if we don't have the specific equipment we need to cover your experiments, we're always happy to get it. Okay. So next I wanted to showcase the experimental expertise in terms of injections that we can offer to our customers. So here I'm showing 5 types of injections spanning from intravitreal to retroorbital and subretinal. We have a 90% success rate for these, and I'm happy to show some data as well. Here you can see the fundus imaging of the untreated eye with no fluorescence. And here's one of our AAV2 injections. You can see really nice labeling in the flat mounts here. And here's another capsid AAV8. I think the 100x is especially beautiful. Um, yeah, I think we like to offer the full gamut of detection items for both mice and rats, and all of our machines are specifically for small animals, so we get really good resolution. Um, I'll show some examples of the data here. Here's some fundus imaging. We can see OCT. Um, this is a retinal OCT where we measure the thickness of the retina. We also have the slit lamp microscopy. Um, so there we can see lesions on the ocular surface. We can image the cornea as well as fluorescence staining in the cornea. Here's some fluorescence fundus imaging after our laser-induced CNV model. So this is showing the hypervascularization in response to imaging, both in our regular animal and our humanized VEGF animal, which I'll speak a little bit about at the end. And yeah, I think I mentioned briefly, but we have a full set of ERG and VEP that we use for in vivo electrophysiology. Okay, so besides in vivo detection, we also provide in vitro services. We can do tissue collection. We have a full gamut of molecular assays. We can do pathological analysis and biochemical analysis, including paraffin section and frozen sections. Here we can take a look at the molecular and histological validation approaches used for our ophthalmology at Cyagen. Here we're showing a full flat mount of the retina. Again, this is the CNV model where you can see the spots with the damage. And yeah, so we're showing the multiplexing with the AAV here and the nice two-color imaging for the choroidal flat mounts. We can also do sequential staining for any one of the markers you may be interested for different cell types. We can do H&E staining. Here's the whole eye and just the retinal area. Here's the cornea. And we also offer immunohistochemistry and immunofluorescence. Um, Furthermore, for the biochemical analysis, we can provide services like Western blot, so we can test for protein. We have qPCR and digital droplet PCR, as well as ELISA to detect expression levels of your target genes. And in the middle here is an example of the digital droplet PCR, and here's some data from the flow cytometry, so we can offer those services as well. And this slide, this is, I guess, an important slide about the genetic models that Cyagen has as well. So this is an organization of all the ophthalmology kind of disease types that we're working on. So retinal degeneration, macular disorders, corneal disorders, glaucoma and intraocular tumors, and then vascular disorders. And we've organized this. So at the top is our Hugo models, and Hugo's our newest platform, and I think the one maybe we're most proud of, where we're humanizing the full-length gene in the endogenous locus. So for that, we're replacing the mouse gene, in this case, the rhodopsin gene for retinitis pigmentosa. And from there, we can make mutations known to cause diseases in humans. And I'll speak a little at the end about one of those disease models that we have called the P23H model. We also have other genetic models listed here, which include knockouts and conditional knockouts. Cyagen has previously created a catalog of cryopreserved sperm where they've knocked out or conditionally knocked out almost every gene. I think they're up to 16,000 with more coming. So we have both global and conditional knockouts for all of these vision models. And then we have other inducible models like the ones I talked about today. So laser-induced CNV. For glaucoma, we have the reperfusion, the NMDA, and the other ones we just reviewed. So please take a look at this slide on our website if you're interested in other diseases like Stargardt's or macular degeneration. So yeah, I take a second to talk a little bit about the rhodopsin gene. This is one of our most popular models in the vision space other than glaucoma. So I'd like to just quickly go over this and our model for the wet AMD. So retinitis pigmentosa is also a chronic progressive disease known to cause blindness in humans. And it's one of the ones with the strongest genetic predisposition. So of all the mutations in the rhodopsin gene, the P23H mutation accounts for 12 to 15% of the patients that we see. So we started by making a humanized model where we wanted to have the full-length human rhodopsin gene in there to assist with drug discovery. And what you can see here is that model, which we call the wild-type humanized rhodopsin model, as well as the rhodopsin model with the P23H mutation known to be disease-causing. And here in the OCT images, you can start to see the thinning of the retinal thickness, which can be summarized in this table here. So both the retina and the outer nuclear layer show decreases in comparison to the control, where the humanized wild-type version shows exactly the same, no significant difference from control. And that's exactly what we wanted to see. So there's no noticeable alterations in the morphology of the humanized wild type, but we do see significant changes in the human disease model. So this indicates that our mutant mouse is suitable for this kind of disease research, and I think provides a more suitable control because here we're comparing our disease model versus the humanized, and the human and the wild type look the same. So yeah, the first step is making the wild type, and the second step was making the disease model. We've now validated both of these models and they're in use in several projects from our customers. We can see here in the ERG that the humanized and wild type show no difference. And we see a drastic difference of the P23H, both in the A wave and B wave, and for the overall flash VEP. We also tested a ASO drug for efficacy on this model. This is one of the standard of care drugs for humans. And we see that changes in both retinal thickness and the ERGs showed the therapeutic effect here of the ASO drug. So you see like almost a half reduction here and also in the OCT images. So we have a super detailed data sheet on this mouse. If you guys are interested, you can find it on the website or email me for it. Um, well, here's a little more of the breakdown over the different days of the ASO treated mice. And so compared with the PBS treated eyes, we see uh, improvement in both the scotopic A wave and B wave. Okay. Finally, I wanted to introduce our humanized VEGF mice. This is a transgenic model we made a few years ago, where we overexpress the human VEGF, creating an animal model of retinal neurovascularization. So it's similar to the laser-induced CNV model I showed you before, but in this case, it's a genetic model that's causing this disruption to the vasculature. And we also see that they have some changes in the ERG, but it's not significantly different here. But our molecular detection, you can see here in our wild type animal, you see no human protein. But in our transgenic animal, we do detect the human protein, which is causing this disease progression. And in this case, the model has both the wild type and our transgenic model have the mouse VEGF. So unlike the Hugo model, which I showed before for rhodopsin, where we replace the full-length gene, this is a classical transgenic model where we overexpress the human isoform by random insertion. So this is a really nice model. It shows through the pathological analysis increased vascular proliferation, which we saw before, and you can see again here. This is IB4 staining, which shows vascular endothelial cells. Um, so if you're interested in our glaucoma models, the RP models or the VEGF models, please contact us. We also tested a standard of care drug on this. Compared to PBS, you can see that the mice who were fed the um, drug had a significant reduction in the vascular leakage, and you see over time, at 28 days, it almost looks totally healthy. So we know that these animals respond to the therapies, and we're very happy to co-develop this for your needs. So I think in the interest of time, I'm going to stop here and thank you for your attention. I'm very happy to take questions.
Thank you very much for the overview, Joe. And glaucoma is a potential eye blinding disease that affects 70 million worldwide. But during your presentation, it almost shocked me how little is known about the genetic cause of the disease. And there's a lot of questions remain to be answered about the molecular mechanism causing the high ocular blood pressure. So at Cyagen, our aim is to provide the researchers the translational models, including the Hugo platform that Joe had just introduced, as well as the CRO capabilities, with the goal of better understanding of the disease mechanism and to develop new drugs regardless or drug modality. And Joe, during your webinar, I've collected quite some questions from the audience. Can you take the time to answer those ones?
Yeah, absolutely. It's my pleasure.
Yeah, so first of all, I think most audience got this question. What's the mechanism that regulates the intraocular pressure?
Yeah, that's a good question. I think I mentioned in one of my slides the main driver for increases in intraocular pressure are the production of aqueous humor and the drainage of aqueous humor. So I think, the balance and equilibrium between those two things are what is pathological in glaucoma. And understanding, where that's caused, whether it be the abnormal production or a decrease in flow rate, is really where we're going in terms of understanding how to kind of turn the knobs on both of those two things.
Great. And in your webinar, in the presentation, you talk about the gene therapies. So what do you think are the advantage at some of the shortcomings of the gene therapy for glaucoma research or therapy compared to the traditional ophthalmology drugs?
Oh yeah, that's a great question. I guess traditional glaucoma treatment strategies primarily focus on controlling intraocular pressure. The gene therapies, though, we can modify and regulate the production of genes involved in the physiology, in the survival rate, and the grow back of, hurt retinal ganglion cells. So it holds promise both as a like alternative approach and a supplementary approach to the current drugs. And I'd say, once glaucoma has progressed to the point where the eye is damaged, we do need to have our eye on like how to address the neuroprotection factor, not just if we see the intraocular pressure go up, we can control that. It's like once the damage has already been done, how do we fix that?
Great. And my last question from the audience is that you talk about quite a different some animal models, and what are the pros and cons of each model, and what is suitable for a short-term study versus a long-term study?
Mm. Yeah, that's a good question. And I think the short versus long term is a good distinction there. So the choice of animal model, I think, always depends on the specific research goals and the like objectives and the time frame in which you need to achieve those. So I'd say the laser-induced CNV model is super fast. Like we do the damage and it increases the intraocular pressure and we can provide the drug treatment, but those increases don't last very long. So those are appropriate for drugs that act on the short-term kind of damage. That'd be the same for the ischemia reperfusion model. And then for models that are better for long term, I'd say the microbead injection is really good. And also some of the spontaneous models, those are the ones I think that best replicate the human progression because you see it kind of come on and we can screen out animals who don't show um, the the progression of the disease that we're kind of looking to treat. So yeah, I think in summary, the genetic and magnetic models are good for long term, and some of the injection models are probably better for short term.
Yeah, I totally agree. I think it just in general, a model or a mouse model can be as best as what question you're asking, right?
Yeah.
Because even for the spontaneous DBA tool, there's still questions on like why DBA is more susceptible to
Mm-hmm.
this glaucoma versus some other inverse strains.
Mm-hmm.
So, Joe, my last question actually from myself is that I know you have been to quite a few ARVO meetings, and any takeaways that you wanted to share with the audience from your latest participation in ARVO?
Yeah, absolutely. I mean, ARVO is one of my favorite meetings, and so I'm a yearly attendee both there and at the SFN Society for Neuroscience meeting that's coming up. So, I yeah, what I've been excited to see is more translation of treatments from mouse models to humans, and so we're partnering with a few pharmaceutical companies. I think, what's really exciting about gene therapy in the eye is it's separate from the brain. Like when we're doing gene therapies for liver disease or heart disease or neurodegeneration, we're typically delivering those systemically, and we have to worry about off-target effects in the liver. But for the eye, the fact that you have the anterior chamber like separate from the whole rest of the nervous system allows us to be a little bit more aggressive about which treatment strategies we can do. And I think, it's really exciting to see what people are doing. There's been retinal prosthetics are starting, optogenetics in the eyes in humans. That's on the frontier. And I think some of the things we showed today, like the work we highlighted for the AAV treatments of regulating genes in the ciliary body to control aqueous humor production, that's like a home run. And I think that we lose sight sometimes as basic scientists that like what we're doing in the lab is really translating to human health. So for me, that's the most rewarding. And I think with the combination of the humanized models coming out that have human genes for our ASO and AAV treatments to like attach to, that's going to help with translation and also just building a community of like-minded people who are solving these problems first in the eye with the idea to go to the rest of the body next.
Yeah, I fully agree. And myself is an immunologist by training, and eye is thought to be an immune privilege site. And so the ophthalmology studies or drug discovery has been sort of at the forefront of those gene therapies.
Mhm.
And I fully agree on the importance of translational models. For example, the Hugo models that we are developing really help to provide the tools that are needed for developing ASO and AAV. So. And so that concludes our today's webinar. And thank you again, Joe, for joining us today. And thank you, audience, for joining us. And see you at next episode. Bye-bye.